Hey everyone, welcome. Before we get started, this would be a really good time for you to silence your cell phones since we're like this little small group. I don't know if you want to be that person that has your cell phone going off. Um, Good morning, good blustery, rainy morning. Thank you for coming out for our faculty talk for the Spring Union weekend. You are at Health Innovation with Purpose, Tufts-Driven Advances in Bioelectronics and Diagnostics with Samir Sonkusale from the Tufts School of Engineering. I'm Marianne Hunt, and I'm the Director of Lifelong Learning in the Office of Alumni Engagement. And in my role, I have this great pleasure of working with our faculty across the schools to develop programs like this one, as well as faculty webinars and as hosts on our Travel Learn programs. We often hear from you how you still want to learn, you still want to be kept up to date on the great things that are happening at Tufts. And really, when you're back on campus, that's the best time. And no better way than to highlight some of our great faculty who are doing just really impactful, amazing things. We have left a little bit of time afterwards for some Q&A. So it'll be a little bit like being in the classroom again. So you get to experience class in the JCC building here, which is great. When we get to that point, I know we're a small group, but if you could just wait for my colleagues to get to you with the microphones, that would be helpful because we're also live streaming this and have a virtual audience. So that'll just ensure that everybody hears your great questions. And now I'd like to say a little bit about Dr. Samir Sonkusale. Dr. Samir Sonkusale is professor of electrical and computer engineering with joint appointments in biomedical engineering and chemical and biological engineering. He is an internationally recognized leader in bioelectronics, biomedical devices, nanoscale sensing, and point-of-care diagnostics. His interdisciplinary research group develops groundbreaking technologies from flexible wearable electronics to ingestible lab-on-a-pill systems that are transforming how health is monitored, measured, and treated. His innovations, including smart bandages, smart sutures, and painless microneedle drug delivery systems, have led to over 40 invention disclosures, multiple patents, and the launch of several startup companies. He received his master's and PhD in electrical engineering from the University of Pennsylvania, and received several awards, including the National Science Foundation Career Award and the Distinguished Alumni Award from his own alma mater, Birla Institute of Technology and Science in Pilani, India. He has also been a visiting professor of medicine at Harvard Medical School and Brigham and Women's Hospital, and also at the Weiss Institute at Harvard. Recently named Tufts Distinguished Innovator of the Year, he continues to advance technologies that address urgent public health needs. His current innovations include a project to develop quantitative pain measurement tools to reduce gender bias in treatment, and research to create rapid point-of-care diagnostics for inflammatory biomarkers. In this presentation, Dr. Sonkusale will share insights from his pioneering work that's at the intersection of engineering, medicine, and entrepreneurship, highlighting how novel devices move from concept to clinic to real world impact. So please give a warm welcome to Dr. Samir Sonkusale.
Thank you so much, Marianne, for a generous introduction. I really appreciate it. It's so nice to be here. I don't know how many of you have taken classes in Halligan, which is right next door, right? So I can see some familiar faces. I don't think I can put names next to the faces because I don't recall, but glad to see you all here. And I want to update you on some of the things we've been working on in Halligan Hall, right next door, on what I would like to call engineering of health, precision health, which is sort of why we are doing advances in bioelectronics. These are some of the pictures that you see are some of the platforms or devices that we have built in the lab over the years. And I'll just take the opportunity to just take you through the journey of the kind of things we've been building in the lab. If you know who my background is, I'm an electrical engineer. But when I started my faculty career, I really wanted to see how engineering can influence or impact or address problems that are at the boundaries of disciplines. It's a lot of the problems that we are looking at, especially in health, in climate change, environment, they require very interdisciplinary solutions. So when I started my faculty career at Tufts, I basically took it upon myself to see how I can help contribute. And so my journey as an electrical engineer, and you will notice in my slides, sort of touches upon many different disciplines: biology, biomedical engineering, chemical engineering. And my lab is sort of a microcosm of the entire Tufts sort of ethos. It has students from chemistry, postdocs from biology, chemical engineering, and so on. And so together, we basically are looking at addressing problems. So my lab's motto is problem solvers without borders, right? So we basically look at problems and then try to come up with solutions. Okay, so before I go, I have to show you a theme around the research activities and what is the theme around which the research activities are built. And it is the idea of closed loop precision medicine. And this is very different than the precision medicine you know, right? So the idea here is that we want to be able to monitor your health and then take targeted interventions, right? And so it's not just looking at your DNA and just prescribing medicine. If you look at how the medicine is practiced today, a doctor would prescribe you a drug or a treatment, not really personalizing to your metabolic state or your age or your gender. They'll probably just do it at a very coarse level, right? And so the idea here is we want to take the smart watch even further, right? And so be able to monitor not just the vital signs, but also the electrolytes, the metabolites in your body. And your health is not just you, it's also the environment you live in. So monitor your food that you eat, your air that you breathe, the environment. So it's like sort of looking at a very holistic view of you and the halo around you, which includes all the environment and the diet and the nutrition as shown on the right side. And with that information, you want to be able to take some targeted interventions, and the interventions could be in the form of a drug, right? That is basically tailored to your health. But I want to do this as more preventive, as more, before you basically get sick. So that should also mean targeted diet or, nutrition that would adapt to what your sensors that you are using to monitor your health are telling you. And so in some ways, if you remember engineering or undergrad course in any of that, there's the notion of closed loop feedback, right? So we sense something and then we actuate and we close that loop with some sort of data processing or machine learning. So it has three pillars, the activities in my research group: to sense, to treat, and then to have some sort of closed loop intelligence. And so what I'll show you are the kind of projects that sort of have this theme in it, in my research lab. So these are some examples. I'll talk about smart threads, which basically have been used for monitoring sweat, monitoring stress, look at inflammation, monitoring both physical activity and cognition, even the microbiome, the microbes that live inside your body and how they impact your health. And I also mentioned food and diet. So you can see there is just a lot of activity in the lab, and the theme is the same, right? Being able to sense and monitor the health and the environment so that we can take smart actions or targeted interventions that would actually improve our health. So all this started many years ago when I had a project funded through the National Science Foundation, and I had a chance to do a sabbatical at the Harvard Medical School at the Wyss Institute to look at how what I was doing for electronics could be used to impact health. And one of the problems that came to us was wounds that do not heal. So if you have diabetic wounds, they have challenges in healing process. And so we teamed up with a lot of collaborators here listed below from Purdue and Mass General and Harvard Medical School to develop smart bandages that actually are the same principle, closed loop precision medicine, where the bandages will monitor whether the wounds are healing, whether there is infection, and then deliver treatment only if needed. right? So if you want to deliver antibiotics, you don't want to just dump antibiotics on the wound because you have antibiotic resistance, a challenge, that we are all suffering from. So you want to be stewards of that, making sure that you only deliver antibiotics when needed. So what you see here are many examples of bandages developed in my lab. This was an undergraduate student when the project started many years ago, which monitored wound oxygenation. And we developed many more for monitoring pH, inflammation levels, so on and so forth. These are flexible bandages that actually go onto the wound. They have sensors, which are electrochemical sensors or optical sensors that convert that biomarker information into electrical signals. And then you can use that information to trigger delivery. So this was a bandage that actually triggers drug delivery wirelessly controlled many years ago. And then we closed the loop, right? So we had sensors in that, and we also delivered drugs. So even the NIH director at that time actually blogged about this. And why was it important? Is because of the fact that this is the notion of medicine that he was also envisioning, right? Where treatment matches your current state, right? How you're reacting, how you're responding to it, right? And so have some sort of a closed loop feedback. While we were doing this, we realized, there needs to be more innovation to be had, because bandages cannot solve all the problems. They are soft and flexible, but they do not go deeper into the wounds. That's where the information is. And I have to say the eureka moment in the lab came when we realized, why can't we make sensors that can be made not on flexible 2D substrates, but on one-dimensional substrates like threads, right? Why is that? Because a surgeon or a doctor can literally take a suture, we call these smart sutures, go inside wherever you want these sensors to be, right? And so that way you can go deeper into the tissue and do a lot more interesting monitoring. So this literally took us on a journey where I will show you a lot of thread-based, you can call it textile-based, so they're more individual threads that are smart. You can then assemble them into textiles to do sensing, to do drug delivery, to do even microfluidics, and also develop electronics. We even made transistors on thread that actually allow us to do this work. So this is just sort of a conceptual vision of what we have: thread-based sensors that can do both physical and chemical sensing. We have things that actually can also do microfluidics that can take the wound exudate, bring it up, and then you can sense it. We can also do drug delivery. So this is showing you a textile patch. What you see there is a real patch on the top right. Each thread is coated with a different drug. So you can actually deliver and modulate the drug profile wirelessly using a phone as to how much drug can be delivered into the bandage. So I still think this is ahead of its schedule in terms of where we are today, but this is a technology that we definitely developed and protected at Tufts. So I'm just going to go through now some examples, right, and of other projects that we worked on. Once we started developing these threads, we realized there's a lot of potential beyond just smart bandages. That's what started the project in the lab. That's what was the seed for all the creative ideas, but we actually basically spawned out into many different areas. One of that was doing sweat sensing. So we took the same threads to see if we can monitor biomarkers of health in sweat. So sweat contains a lot of very useful biomarkers. And these biomarkers could be your diet level or nutrition. Let's say you don't have sufficient vitamin exposure, or you don't have proper nutrition. You can actually monitor that by looking at your sweat. And it has a lot of these information. So you can actually look at sweat for all this. So what we did was we took the same threads that I showed you just a few minutes ago about what it can do, and use them as a part of a Band-Aid to monitor electrolyte levels, right? So you're in a classroom, I have to show you some plots, right? So some plots just to basically show you that it can monitor ammonium, it can monitor sodium, it can monitor pH, it can monitor lactate. Lactate's a very good marker for if you're an athlete and you're training and you want to monitor whether you cross the anaerobic threshold, right? Am I functioning at the right level? You can monitor lactate. if you haven't taken enough water or hydrated, right? I mean, you want to monitor all the electrolyte levels. A lot of potential using these threads. As I say, innovations never follows a straight path. Right? There's always these different branches that spun out of the same thing. And there is one more that came out in the lab. Why threads? Why can't I just put directly the sensors on the skin themselves? Why do I still really need, if I'm just monitoring sweat, why can't I just put the sensors directly on the skin? Skin is the substrate, right? And so that brought us to sweat sensing tattoo. This is brand new sort of research activity in the lab. So, and this was funded through, at least the funding started through some projects where we were interested in looking at post-traumatic stress disorder, chronic pain, cognition, sort of spanning many different areas of research at Tufts and beyond. And so, as I mentioned, these are the areas. So we want to monitor neurotransmitters, electrolytes, things like that using sweat. So we made tattoos. These tattoos, each one of them are like threads. They have the same chemistry, but they are basically imprinted on your skin. So just like a tattoo that you put on the skin, each element there or each electrode actually is monitoring. Interestingly, this tattoo also powers itself. It takes the sweat, moisture, and powers the sensor using the moisture. It does not have a lot of power, which means it's going to transmit data very infrequently, but it doesn't need a battery at least, right? So you basically have a very simple platform. So this is a tattoo, how it looks. This is an example of a student running the tattoo on the skin, some sort of data. And what we like about that is flexible, self-powered, user-friendly, biocompatible. So this is another innovation out in the lab, again, building upon our thread-based portfolio, which is beyond sweat, there are other biological fluids that are non-invasive, right? And so saliva is another source of very useful biological fluid that has a lot of useful biomarkers for your health and wellness. So, and we had a project where we were very interested in monitoring stress during learning. This was a project-funded collaboration with our Department of Psychology and Childhood Education, where we were interested in learning how stress affects learning, and then how can we improve that. That's how it started. There are other projects in the lab as well that use that in post-traumatic stress disorder in adults and so on. But the idea is, can you monitor stress, right? And so that's how we started doing this. And we realized that we can actually do that by monitoring cortisol, which you may know, some of you may know, is a very good marker for stress. But there is a challenge in monitoring stress, right? What is the challenge? The challenge is, if I ask you, this is a sensor, please test yourself for this marker for stress, you might stress out just by the thought of getting measured. So we wanted to find a way to embed monitoring, health monitoring, in your day-to-day life, right? And so something that you can use on a daily basis. And so we were thinking about, okay, how do we get to saliva, right? I mean, and so then we realized that flossing, I know not everyone flosses all the time, but flossing is one of those activities where you can embed the sensing into the floss and at least reduce the barrier to monitoring things which are hard to measure, like cortisol or saliva. So can we monitor stress? So we do have this. This is actually now one of the things that has spun out of my lab called SensiFloss. And it does monitor a lot of different biomarkers now beyond cortisol. So there are a lot of other hormones in saliva that we can monitor using this platform. So this slide is just to show you that beyond threads, we also work on something called paper-based devices. Just the innocent paper that you use for writing, right? I mean, that paper can be engineered and be functionalized to do a lot of sensing. And this one actually monitors inflammation markers, right? And so the chemistry on this allows you to do monitoring of cytokines like IL-6, CRP. CRP is the cardiac protein that we would like to monitor for in case of heart challenges. TNF alpha, which is another cytokine. And what is innovative about this platform is basically it's a very sample to answer diagnostic device. You put the sample in, each of those channels that you see on the paper correspond to different analytes, right? And so the reaction length on that channel tells you the concentration of that marker. So it's very simple to do. It took a while for us to develop this. We call this distance-based device because it's a length or the distance of the reaction. And interestingly, there is no electrical engineering involved in this. It's more chemistry, and I'm an electrical engineer by training. So innovation, as I said, can take you many different paths, and something, as curious learners we are, we basically make sure that we follow all those very interesting pathways here. I want to talk about this recent project. This is ARPA-H is a new funding agency that had a program out for Sprint for Women's Health. And the idea was to tackle another challenging problem, which is chronic pain. Right? So chronic pain disproportionately affects women than men, mostly during perimenopausal period to postmenopause, right? During that period. It's not the only thing, but it is one of the symptoms that actually has a lot of underlying pathophysiological causes, change in hormones, things like that. And one of the things the ARPA-H went out to do was to see if there was a biomarker. Is there something in you that we can measure that can quantify pain levels, right? And there is no universal single biomarker for pain, right? So, and at that time when this project started, I was working with the Henry Jackson Foundation, the Uniformed Services University. This is a military hospital in DC. And they have this huge biobank of samples for both women and men and people who have. And so we wanted to see if we can mine those biomarkers to see if we can find some analytes. And I have to say that I wanted, we could find a panel of biomarkers that collectively together can provide you with a fingerprint or some information about pain levels. So how are we doing it? We have these microneedle patches that actually go in. They do not cause any pain. They go just under the skin. They acquire interstitial fluid. Interstitial fluid is another non-invasive biological fluid, just like the sweat or the saliva, but it's very close to blood in terms of the concentration levels. And then you put the sensors on these microneedles that can actually measure this multiplex panel. So the data is coming, but I just wanted to get to you an overview of what's happening in the lab. So this is how the platform looks. These are the sensors. And this is our little readout device called PainSense. So you put the sensor in. And the clinical trials are going on right now at Northwestern Medicine. How much time do I have? Okay. So I'm open to questions. If you have any in the middle, I can take any from the audience, or I can wait till the end. Yes, go ahead.
Do you have any full patterns for the tattoo? Do you have like a heart pattern or something for the tattoos?
We could make, yeah, definitely. Yeah. Right now they're just, simple tattoos, but we can pattern them in different forms.
So I think one of the previous slides was talking about the floss, the strings. What's like the thickness of it? And how does that compare with the actual, like an actual floss or a string?
Yeah, so the question was, the length of the string and how does it compare with the actual? There is no sensing on the floss. The floss is the same. The sensor is in the handle. So we take the saliva and the thread filters the saliva out, takes all the stuff out of the saliva, goes inside, and inside the handle is where the sensing chemistry is. So nothing's happening in your mouth. So the sensor's far away from that. So I'm going to switch gears, right? So this is threads and thread. I'm going to talk about one more interesting thing. How many of you know that we have more microbes in our body than our own cells? Right? So so a lot of people, we have more microbes. So sometimes we have to wonder who's controlling our health. So probably microbes are driving our health. And there's a very interesting story about how I started working in this, but I'll skip that in the interest of time. But the idea is, why don't we monitor gut? And there's been a lot of research lately where they study your fecal microbiome to find out correlation between the microbiome and disease. They can only find correlation, no causation yet, right? And the reason is they don't know where those microbes came from. And so the project started in my lab where we were very interested in knowing which microbes inhabit the most important regions of your GI tract, and the most important region being the small intestine. That the microbes there, they regulate your immune system, they're important in digestion, and a lot of autoimmune conditions, there is a theory that it is because of gut inflammation, small intestinal, but there is no, it's like the black hole of GI tract. You just don't know what's going on in there. And so I had the story, the abbreviated story was when I went to a program manager looking for funding, which is where I spent some of my time. And they're like, if you can solve this problem, you can come and see me. This was around Thanksgiving, Christmas time. And I was like, okay, let me come up with some sort of a solution. So we made this ingestible device, which is very simple, that actually go in and collects a sample from the small intestine. And so that's the basis of some of the projects started. We did a lot of work on this. This is one of our first pills called the osmotic pill. We can test it in pigs, mice, a lot of animal studies. And right now, this pill or this capsule is being tested at the Tufts HNRCA, which is our nutrition school, as part of a diet intervention study. So, which is a very well-known study. We know that people who are on high-fat diet or high-protein diets, their microbiomes are different. We want to validate if this capsule really works. So we went through all this process of IRB, getting approvals and so on and so forth. So with that, I'm just going to end. I'm just going to show you some of the other research projects in the lab. So we also work with monitoring plant health, which is also looking at food quality and freshness. I already showed you this. We also have microneedles for delivery of drugs. This is actually a company that spun out, one of the first companies that spun out of the lab, called Anodyne Nanotech. We can also monitor your environment, looking at pollution, detect sleep disorders. And I'm a circuit designer, so I still design chips, all the time for miniaturized instrumentation. So these are just to show you some companies. I wanted to show that Tufts innovations, Anodyne, Microvitality, and right now saliva sense or pain sense is coming out from the lab. Thank you.
Great. Thank you so much. Plenty of time for questions. And Paige and Stephen both have microphones, so feel free to ask away.
Hi, Professor. In terms of challenges when you're designing these, I know there's a lot of different projects. What were some of the biggest challenges you had to overcome to get to these achievements?
Very good question. I know Usman. So the challenges, I would say they are not more, I understand there are challenges, but the challenges mostly happens in interdisciplinary research is to be able to talk the other language, right? So when you're interacting and you're doing work that are the boundaries of discipline, to be able to do, where if you're talking to a clinician to understand their real needs and how it translates from a problem to a specification that you can actually get your head around, right? And so I would say that conversation or communication is probably the biggest one when you're looking at, and the engineers are very capable of solving a lot of problems, right? Scientists or engineers, they're very capable of problems, but sometimes I've found over the years that asking the right question also matters, right? And so as I was doing this research, we were looking at, okay, what are the problems we really want to solve, right? Where is real value in terms of where the impact can be? And so that's sort of the second aspect of the work. So it's not a challenge so much as what is your vision or what is your guiding light behind? And it takes a while sometimes to figure out, is this really worth the effort that you're going to put in? And I have to say, Tufts is very nice that way. There are less silos between departments. I have students from other departments that are part of my lab. When I started, we only had electrical engineers, but right now my lab is half chemical engineers, other half electrical engineers, postdocs in chemistry and biology. And I love it. That environment is just amazing in terms of addressing that communication problem, right? Because now we have people who can understand what the other language is to tackle the problems.
With the, say, the chronic pain metrics or gut metrics or whatever biomarkers are identified, what actions are then taken from there? And is that something that's more handled in as part of this research or in conjunction, or is that more of a separate area that's then kind of passed on to?
Correct. So you're talking about the gut health, the microbiome work. It's so early to know. So the treatments for a lot of GI disorders, for example, people who have GI issues will tell you that they go through many years of trying different treatments and nothing seems to work, or they just don't know what's working. So I don't think they have an answer to that question for everyone, right? And the reason for that is they just don't know enough about that area of this. So we was addressing the problem of getting there inside and to figure out, okay, how does microbiome influence your health? And then what about the small intestinal metabolites? And so I am a tool developer, right? Technology developer. So I want to make this pill as accessible to as many scientists and researchers who are doing fundamental studies on understanding the pathology of these different diseases and see if they can then come up with more targeted treatments. So we're very early on, but I have a very strong feeling that this will help sort of address all the small intestinal links to the health conditions. But that's where we want to go eventually.
Thank you.
What are some of the materials that you're using to build these sensors in regards to like the wound healing sensor, also the sensors for hydration? I was curious to hear about the substrates as well as just even the sensor itself.
Correct. So very good question. And that's why we like textiles, because the wound bandages are all textile bandages. If we embed sensors into the bandages themselves, then we're not introducing some new synthetic material that have not been tested in terms of biocompatibility or toxicity. So the threads let us work with our cellulosic materials or these materials that have been around for many years that are already part of the treatment, make them smart. Is the approach there, right? So we basically embed sensors on them. We do put some electronics on them. In smart bandages, we've been very careful about how we interact. We typically tend to take the wound exudate out on the surface of a bandage where the sensors are. So the wound exudate is just basically plain textile threads, and all the sensing is essentially sitting on the surface. So there's a lot of engineering and a little bit of tinkering there, where most of the materials are, I would say, biodegradable, well-known materials that people have already worked with.
So just kind of understanding how you came to that approach, right? Like, okay, here's what we're going to do. We're going to use a biodegradable material and, so on and so forth, right? So what was the thinking? Like, what was the context behind what led you to, go in that direction is what I would love to understand.
So it's a very iterative process, right? So if you're thinking about how the innovation, I can point to a few things that happened during that smart bandage work. When we knew we could do flexible electronics or flexible sensors, right? And the materials that everyone uses for flexible is some sort of polyimide or one of these very simple plastic sheets. But nobody uses that on the wound. So when we were innovating and we're talking to people, we realized that it's nice to publish a paper or have an idea around that, but it's not going to fly because it's not a very common material to use. And probably the reason is it's probably irritates the wound, it's not biocompatible, things like that. So you already start thinking about some other materials, right? We need to start innovating, thinking about So if you're flexible about this, that you have to get to the bottom of the solution, you really need a solution, then you say, okay, what are they using now? Right. So if they're using bandages, and we are already working at that time on these textile threads or materials. So we said, okay, let's just make the bandages with these textile threads as substrates and figure out a way to transfer the same chemistry that we were doing on flat plastic substrates onto that, right? And so you start going there, and suddenly you realize, okay, I can actually do more than just bandages now, right? As I said, it's sort of an iterative process sometimes. It's not as if the solution just comes directly, right? It's as I said, it's a journey. You have different branches around the common theme or a common strand, and it takes a while to get there, but a good team. I have a lot of really great students in my lab and postdocs, so quite creative.
Thank you.
You have a wide range of projects in your lab. I'm curious how they come about. Are students involved in the ideation of these projects? Do you have external partners that you work with that bring problems to you, or is it something else?
It's a combination of all of those, right? And so, the lab, essentially, when you come to the lab, and we are most of the times PhD students, they're sort of funded on a project, right? And so we start working on it, but they have freedom to think independently about, and I do insist on finding a problem that's meaningful, something that actually that you can solve, something that will add value, right? And so the students do come up with very creative solutions in the lab. And then also the interaction, just the ecosystem, the people that you're talking to. So we had conversations with people at Tufts Medical Center, and we realized, wow, this is a problem that nobody looked at, and looks like we have the tools, right? And so why can't we approach and try to get a solution for this? Just so much to do, right? I mean, my lab, I sometimes call it as a garage. So my lab is right there. You can come by. It has everything from 3D printing, laser cutting. I want to think that if you come into my lab with an idea, you should get out on the other end of that lab with the device in your hand, right? And so that level of interdisciplinarity exists. So in some ways, if you come to the lab, you talk to people, you probably find problems to solve. You have to decide which ones to take, because there's too much to do.
I'm also an electrical engineer, and by my recollection in the studies, we never learned like composition of sweat or microbiome. How do you know what you're doing in that area? Like, how do you get valid knowledge, and where are you pulling?
Yeah, and that's a great question. Again, this is essentially the same. People do ask me that, and I have to say, if you're a curious learner, you tend to try and read and interact with people a little bit more outside, because that's where the interesting problems are, right? And so, as I was saying, in the context of microbiome, I happened to go to a program manager with some other ideas of my own, but had a really long conversation about where the problems were. And I forgot about what I wanted to do to just understanding what she wanted me to do for that particular field, right? And so went back and thought a lot about it, and see if the team can come up with. So there are problems everywhere. We have a lot of things to solve, and then once you get sucked into a problem, you want to read more, you want to interact with a lot of people, see what they're doing, what they're thinking. And so I think the story has been consistently like that. I've just basically had an opportunity to interact with a lot of very smart people who had very interesting problems. And not everything, what you see here is probably 10% of what we attempted, right? So it's good to show the things that worked, but there are a lot of problems that we tried, we worked on, we thought about, and had problems. And so that also comes down to how deeper your understanding is sometimes in some of the areas. But that doesn't stop us from trying, because the effort that we put in definitely will make an impact if we can even solve 10% of the problems that we have. So I don't want to say that everything is, once you get the problem, you can get to it, but you can attempt, right? And so that particular aspect is also the discovery part, the creative part of that. And that requires learning. And sometimes we know a lot about the problem by reading and talking to people, and sometimes we don't know enough.
Had a quick follow-up question. Maybe it's more strictly to the EE side of things. So what I notice here is there's a lot of different applications, right, for sensor technology. If we stick to the core sensor technology, if you look fast forwarding about 30, 40 years, right? Where do you see that going? Like, sometimes maybe in my naive impression of the industry, it's almost like you could potentially draw a parallel with what happened in the semiconductor industry, where you might have, certain limited area, you do one sensor, but now if you want to do, let's just say, enhance your diagnostic abilities and monitoring abilities, you might want to fit 10 sensors for 10 different purposes. Now real estate becomes an issue, then miniaturization becomes an issue, and there's a lot of other things that could become an issue. That's just one side, right, of things, but there's so many different angles that we could go. Where do you see this going in 40, 50 years time?
40, 50 years sensing.
Yeah, specifically like your sensor technology, what's the direction it could be moving towards applications, that kind of thing.
So, I like to think of this as how much do you know about yourself, right? And so in 30, 40 years, I'm envisioning, I'm not saying the solution exists, know everything about everything that's happening inside your body, to neural activities, to metabolic changes, to being able to sense and monitor every biological pattern, right? At the resolution at which current chips are so small, there are billions of transistors on a single chip, right? We started 1960s or 70s with 5 transistors to billion. Imagine billion sensors in the body, being able to monitor every little detail. And why? Is because we want to understand our health and how everything impacts, not just that, but just provides us with more understanding of life in general, right? And so that is sort of the vision for a lot of people who are developing a lot of sensors, continuous real time at all scales, molecular scales at single cell level, all the way to organs, all the way to tissue, to humans. And I'd like to call the halo around you, right? So the environment, how does that all impact? So can I monitor at scales longitudinally and at that spatial scale? How much of a data that you can generate, right? And so I think that's where I would say, my vision for monitoring is. And treatment is if you know this much, and then you basically then know where the interventions are, right? So you need to know first before you can do targeted intervention. And then you know exactly how this drug is going to affect you, or how this treatment is going to affect you, because you have that level of information.
Hi, thank you so much for the talk. I'm a clinician, I'm a physician, and so these are some really hard problems that medicine has been scratching its head at for years, like pump pain and microbiome. My question was more, I think somebody touched on the medical school. I was just curious what interaction they have in terms of like the clinical applicability and testing for some of the devices that have been mentioned.
So though the regulatory framework is basically where you're getting at in terms of and how does it fit into the standard of care. And so it's amazing how engineers, we come up with solutions, but we do not look beyond the problems that, how do you translate them to the actual? And I'm learning a lot. And some of these technologies like our pill found its way into human subject trials. I want to see that. And if you look at the device that finally is ready for human subject trials, it is the simplest device. It has absolutely no complications, even though we made so many different ingestible devices. And I have suddenly realized that innovation can also be guided by that process of knowing what will go in. But you don't want to stifle innovation at the research lab by not allowing yourself to dream beyond that. But when you get to that level where you have that clarity that, okay, you've come this far, what will now fly? It's important as researchers to also start thinking at that point in time or before that, I need to see some of this out there. So what aspects? So I completely get where you're coming from. Not everything is going to find, but we still have to start innovating and doing things. Because if we don't show what's possible, at least at our level, then we will stifle innovation. So I want to keep the the open curiosity and the questioning and coming with solutions still, and all the students, they're very creative with their ideas. And so I don't want to say that, you can't do all of it, but you're right. You have to have a little bit of the process, and bringing that into the curriculum at some point would actually definitely be helpful. I don't know whether I got to the answer to the question.
Thank you, though. I appreciate that.
Hi, so I'm not an engineer at all. I'm actually an attorney. But I'm curious, as you talk about collecting potentially billions of data points, do you worry about the privacy concerns or anything related to that and having that much data, especially if it's going to be so easy to collect through these small sensors?
I'm already worried about LLM collecting all my data about all the things. Definitely worried all the time about all this privacy issues. There's a lot of effort that needs to go into security and privacy. So that closed loop path that you see, that central controller, the brain of this closed loop, really needs to be secure, needs to have. And so that needs to have the ability to not just sense and deliver treatments, but to be able to encrypt signals, keep the privacy in mind. And there are all the other factors. You have to also think about socioeconomic and all of that. Those considerations need to be there at some level. So great question. I don't know whether I have an answer to that, but a great question.
Other questions at all? All right. Well, that was wonderful. Thank you so much. Every time I hear from not just the biomedical engineering area, but some of our other great centers, it's just mind blowing. I think when we first talked, I was thinking about science fiction and Star Trek and how they can just scan for everything. So just really great and proud to have you here at Tufts. And thank you for your time.
Thank you.